In this video, we are going to look at a detailed explanation of the electron transport system. An electron transport system refers to a series of protein complexes and other molecules that are embedded in the inner membrane of a mitochondrion. A cell uses this method to gather the energy into electron carriers, that is NADHs and FADH2s, and then transform that energy into proton gradient that is then going to power ATP synthesis. From the cell biology, we meet a certain cell organelle whose inner membrane is highly folded. So our cell organelle is known as a mitochondrion. A mitochondrion has got various parts. So it has got what we call the outer membrane. It has the inner membrane. It has the intermembrane space. And then it has got what we call the matrix. I would like us to use the mitochondrion to explain what I have just drawn here. The matrix corresponds to this particular space down here. Then we have the intermembrane space, which is here. Then we also have the inner membrane. So the inner membrane is highly folded to form what we call crystal. So what I have drawn here is the inner membrane of a mitochondrion. Then we have the outer membrane, which is up there. On our diagram, we have protein complexes labeled 1, 2, 3, and also 4. We also have ATP synthase. Protein complex 1 has got FMN. FMN means flavin mononucleotide. It also has the iron sulfide center. Then we have CoQ within the inner membrane of a mitochondrion. Then we go to complex 2. Complex 2 has got a FAD attached to it. That is a FADH. Then it has got a iron sulfide center. Complex 3 has got cytochrome P, iron sulfide center, cytochrome C1, cytochrome C. Complex 4 has got cytochrome A3. This is oxygen molecule. This is cytochrome A. This is copper center. Then this is the inner membrane of the mitochondrion. Then ATP synthase has got various parts. That is C10. It has beta subunit and alpha subunits. And also we have the gamma stalk. At this point, you need to know the golden rule of electron flow. Electrons flow from a region of high reduction potential to a region of low reduction potential. You might ask yourself what is meant by the term reduction potential. So reduction potential means the ability to reduce another molecule. So a molecule that has got higher reduction potential has got a higher ability to reduce another molecule. That's a very, very important point for you to note. Something else for you to note is of electron flow. So we have direct transfer. Direct transfer. This is seen in iron sulfide centers, where we have iron 3 ion species getting reduced to iron 2 ion species. So when iron 3 ion gains electrons, it gets reduced to iron 2 ions. Method number 2 is as a proton. I believe that at this point you know that the chemical formula of a hydrogen atom can be written as this. The method of electron transfer is as a hydride. Next, let us start discussing about the complexes. So we begin from complex 1. Complex 1 is L-shaped. As you can see it, It has got the vertical component and the horizontal component. So the vertical component is partly held in the inner membrane of the mitochondrion and is partly exposed in the matrix of the mitochondrion. Let's look at the contents of complex 1. Complex 1 has got FMN and iron sulfide center. FMN means flavin mononucleotide. Complex 1 only accepts electrons from NADH through FMN. Due to this, complex 1 is also known as NADH dehydrogenase. But it also has iron sulfide center, and we've discussed that iron sulfide center transfer electrons through what we call direct transfer. Let us look at coenzyme Q. We meet coenzyme Q in the inner membrane of the mitochondrion. When you take a closer look at the inner membrane of the mitochondrion, you will realize that it has got the phosphate heads, which are normally hydrophilic, and the lipid tails, which are normally hydrophobic. So you expect anything that interacts with the hydrophobic region of the lipid tails to be also hydrophobic. So coenzyme Q is highly hydrophobic. So it dissolves in the inner membrane of the mitochondrion and transfers electrons. What makes our coenzyme Q to be highly hydrophobic is due to its long tail of 10 isoprene units. It also has a guanine ring. So let's look at the structure of coenzyme Q. So this is the structure of our coenzyme Q. I've just said that coenzyme Q has got a guanine ring and also a long tail of 10 isoprene units. So it is this long tail of 10 isoprene units that make coenzyme Q to be highly hydrophobic. So that makes it highly soluble in the inner membrane of the mitochondrion. So let's look at the reaction of our coenzyme Q. The role of coenzyme Q is to couple the transfer of electrons and protons. So it accepts electrons from complex 1 through iron sulfide center and gives out those electrons to another molecule that we are going to look at. So let's look at how it reacts. So CoQ accepts 2 electrons from the iron sulfide center. So we just say that CoQ couples the transfer of electrons to that one of protons. I want to draw this one using the shortest form possible. So I will represent it with letter R due to the short space. Due to reduction in the space, I'm going to represent this one with our letter R. So during the reaction, this region will gain the electrons so that it becomes an OH, and also this region will be an OH. So this is our methyl group. So that is OCH3, and this one is another OCH3. So our electrons are going to be distributed. They will move from this point to that point, like that, then like that. Oxidized form, this molecule is known as ubiquinone. But in a reduced form like this, it is known as ubiquinol. So ubiquinol refers to 1,4-dihydroxy-ubiquinone. Summary of CoQ. I've said that CoQ has got a quinine ring and it has got a long tail of 10 isoprene units that make it highly hydrophobic, and therefore it dissolves in the inner membrane of the mitochondrion and transfers the electrons. Another difference is that FMN is made up of isoxazine ring and also ribitol in addition to it. We are going to discuss that one in a lesson known as nucleotides. The names of the compounds are self-explanatory. FID has got 2 nucleotides and adenine is specifically the base. FMN has got a single nucleotide and is not specific to adenine as a base. Next, let's go to complex 2. Complex 2 is also known as succinate dehydrogenase. This complex 2 is a very special enzyme in that it is the only membrane-bound enzyme that is found in the electron transport system and is also found in the TCA cycle. This complex 2 is also known as succinate dehydrogenase. The reason why it is known as succinate dehydrogenase is it dehydrogenates succinate. This is what it does. It picks electrons from succinate. Let's say this is succinate. And it converts this succinate into fumarate. That's why complex 2 is also known as succinate dehydrogenase, because it dehydrogenates succinate. In the TCA cycle, we meet this succinate dehydrogenase where it works on succinate and it converts it into fumarate. Succinate is a 4-carbon dicarboxylic acid that is also symmetrical in shape. It has got the following group of atoms. So I want to illustrate using a different color so that you can easily understand what I'm going to talk about. These ones I want to use black ink. So that's our succinate. So succinate dehydrogenase converts succinate into fumarate. Dehydrogenation occurs between the 2 middle carbons, this carbon and that carbon, so that this is the resulting molecule. We shall have this carbon and this carbon. So we shall lose 2 hydrogen atoms, so that we shall be having only this hydrogen atom and this carboxyl group. So that we only have this hydrogen atom and the carboxyl group, same to this. We shall have our carboxyl group here and our hydrogen atom here. So these ones will be double bonds. So succinate dehydrogenase carries out dehydrogenation of succinate with the help of FAD as a coenzyme. So FAD is reduced into FADH. FAD is reduced into FADH2. That is essentially what occurs in the TCA cycle with the help of succinate dehydrogenase. Complex 3 has got cytochromes and iron sulfide center. So let's begin by looking at the cytochromes. Cytochromes are special protein molecules that are located in complex 3 and complex 4. These molecules have heme groups that confer them a special characteristic of UV light absorption. Cytochromes are named basing on the wavelength of the UV light they absorb. So they are named as A, B, C, depending on the wavelength of light that they absorb. Complex 3 has got cytochrome D, iron sulfide center, and cytochrome C1. It's also attached to cytochrome C that is in the intermembrane space. Contrary to core Q, cytochrome C is highly hydrophilic. So it dissolves on this side and transfers the electron to complex 4. So the role of complex 3 is to couple the transfer of electrons from core Q to cytochrome C with pumping of protons. The role of complex 4 is to accept electrons from cytochrome C and transfers those electrons via a series of copper ions, then through cytochrome A to A3, then to the final electron acceptor, which is oxygen. ATP synthase. So from the word ATP synthase, it means it catalyzes synthesis of ATP. So ATP synthase has got various subdivisions. So we can subdivide it into F0 subunit. That is the first section. Then it also has what we call gamma subunit. So gamma subunit is here. Then it has got F1 subunit, which is located in the matrix of the mitochondrion. So this is F1 subunit. So the F0 subunit of ATP synthase has got ability to spin. It spins once a proton enters it. So when it spins, it also causes the gamma subunit to spin. But when you look at the F1 subunit, it is held by a beta stalk, and therefore it cannot easily spin. So guys, this information can easily be found in the Learning Just Biochemistry. That is the 7th edition, page 1812. So we learn more about this ATP synthase. We can now discuss the electron transfer and the pumping of protons. NADH is going to contribute its electrons to the electron transport system. So it is going to donate 2 electrons to the electron transport system through flavin mononucleotide. Our NADH is going to get oxidized, and it is going back into the TCA cycle or pyruvate decarboxylation to pick more electrons. The 2 electrons are then transferred through a series of iron sulfide center, which are then transported to coenzyme Q. The movement of electrons through complex 1 generates electric current, which makes it to be supercharged. This causes complex 1 to pump 4 protons from the matrix of the mitochondrion into the intermembrane space. Therefore, there is a proton gradient that has existed between the matrix of the mitochondrion and the intermembrane space. Enzyme Q does not transfer electrons directly through complex 2, but it moves within the inner membrane of the mitochondrion and does what we call transfer of electrons. Let's go to complex 2. Complex 2 is also known as succinate dehydrogenase. It carries out dehydrogenation of succinate. So it picks 2 electrons from succinate, converting it into fumarate. The 2 electrons that are picked from succinate are put onto FAD to form FADH2. From FADH2, our 2 electrons are transferred through the iron sulfide center, which then move to coenzyme Q. We know that step 6 of glycolysis is involved in the yielding of NADH. So the question is, how does cytosolic NADH shuttle its electrons in an electron transport system? There is an enzyme known as glycerol 3-phosphate dehydrogenase that is located somewhere here, which is used to shuttle electrons to coenzyme Q from the cytosolic NADH. To CoQ, our electrons are transferred to cytochrome B, complex 3. So there are 2 electrons. These electrons are then moved through a series of ion center, that is iron sulfide center, to cytochrome C1, then to cytochrome C. Movement of electrons through complex 3 makes it to be supercharged. This causes complex 3 to pump 4 protons from the matrix of the mitochondrion into the intermembrane space. Contrary to CoQ, cytochrome C is highly soluble on the phosphate heads of the phospholipids. It is highly hydrophilic. So it dissolves and does what we call transfer of electrons to complex 4. From cytochrome C, the electrons are picked through a series of copper ions, then to cytochrome A3, then to cytochrome A, and finally to the final electron acceptor, which is oxygen. When oxygen interacts with 2 electrons, it forms water molecules. In this case, we are going to react it with half the oxygen molecule. We know that a single water molecule has got 1 oxygen atom. That one implies that 2 protons react with half the oxygen molecule to yield a single water molecule. We have used a single NADH to produce a single water molecule. So that means that we shall need 2 NADH to consume 1 full oxygen molecule. If we used 2 NADHs, we will be having 4 protons and a single oxygen molecule to form 2 water molecules. Movement of electrons through complex 4 generates electric current, which makes it to be supercharged. So complex 4 pumps 4 protons from the matrix of the mitochondrion. But the difference from other complexes is only 2 of those protons reach the intermembrane space. I have shared a mnemonic on how to remember the electron transport system. Check in the comment section. I'm going to share how you can easily remember the electron transport system. We have generated a proton gradient between the matrix and the intermembrane space. So the protons will tend to go back to the matrix of the mitochondrion, and they will be looking for a way of entering back into the matrix of the mitochondrion. There is only 1 molecule that is permeable to protons at this point. So that molecule is the ATP synthase. It's divided into 3 subunits. That is the F0 subunit, the gamma subunit, and the F1 subunit. So when a proton enters the F0 subunit, it spins in a counterclockwise direction. When the F0 subunit spins, This causes conformational changes in the gamma subunit, which also spins in turn. When we look at the F1 subunit, it does not spin, it is intact because it is held by the beta subunit of the beta stalk. When the gamma subunit spins, it causes conformational changes in the beta and the alpha subunit. This causes the interaction of ADP and inorganic phosphate to form ATP, which is our major target in this reaction. So we have generated our ATP, and that means our mission is accomplished. Link about the glycerophosphate shuttle in the comment section.